Hi, my name's Erica Nagy, and today we will be talking about the article titled Gene Therapy Comes of Age by Cynthia Dunbar, Catherine Hai, Keith Jung, Donald Cohn, Kia Azawa, and Michael Sadelain. So the idea of gene therapy utilizing exogenous DNA really came about 50 years ago or so, and originally utilized transfer vectors that were actually found to ultimately cause more harm than good in the human body. Some of these health risks stemming from these early gene clinical trials for gene therapy included induction of inflammatory responses to the vectors and induction of malignancies due to proto-oncogene insertions that these vectors used. And in the early 2000s, lentiviral vectors, genetically modified T cells, and adeno-associated viruses proved to be some of the first successful and safe options for gene therapy. In 2017, the FDA actually ended up approving the latter two options for treatment of both B-cell malignancies and congenital blindness. So currently, the most promising vectors that are being researched are retroviruses and adeno-associated viruses. Retroviral vectors, such as spumaviruses and lentiviruses, were found to be beneficial because they can carry various products of different sizes into the genome, and they have the ability to undergo reverse transcription, which allows them to incorporate the DNA that's desired into the genome, but they also cannot reproduce themselves within the human body. AAVs, on the other hand, are sourced from a parvovirus that is both non-pathogenic and that's unable to replicate in the human body. So they're similar to retroviruses in the sense that they're great vectors for gene therapy. However, unlike retroviruses, they can't carry various sizes of genetic material into the genome. So they're kind of limited in the size of gene product that they can incorporate into the DNA episome. And because of that, scientists really aren't sure how long that DNA episome will last and how long it will live, per se, in cells that are already in the post-mitotic phase. So two different types of virus vectors that are very beneficial, but again, both very different. One can carry a variety of sizes of genetic material into the genome, and the other one can carry a limited size of genetic material into the genome, but they both are non-pathogenic and they both can't replicate in the human body, making them great vectors for gene therapy. So along with these retroviruses and these adeno-associated viruses, which again prove to be great vectors for gene therapy, in vivo therapy has undergone a lot of testing. The main targets for in vivo therapy have been the eyes, the neuromuscular system, and the liver. And ex vivo testing has also been utilized, and that's been utilized in the production of stem cells, which can differentiate into different types of cells that are needed within the body, and also CAR therapies for cancer. So while a lot of the methods that we discussed above have proven to be promising methods for gene therapy, they also kind of lend themselves to a lot of controversial debate, especially since there's many aspects of gene therapy that seem to be understudied, such as CRISPR-Cas9. There's still a lot of debate to whether CRISPR-Cas9 and similar technologies are even ethical or should and can be used on humans to treat human disease. Ultimately, I think this article covered a lot of gene therapy methods that are promising and that prove to be very versatile also in treating human disease, slowing the progression of disease, and ultimately, hopefully, finding a cure for a lot of disease. I think gene therapy is also a really invaluable tool to kind of solving issues within biotechnology, such as adverse reactions to medications, antibiotic resistance, and ultimately the inability to find a cure with the methods that we've been using so far for a lot of human diseases, such as cancers.